All right, good evening, everyone. My name is Sarah Alger. I'm the director of the Russell Museum here at Mass General. I see some uh, familiar faces and some new faces. Welcome all. Welcome back to our lecture series. As you know, we take July and August off. One housekeeping note for all of you who are on Zoom is once Dr. Baer is done with his lecture, just use the Q&A function to ask any questions. And a plug for our subsequent lecture, it'll be a little bit different. We'll be screening a short new documentary that is called White Nine, which takes its name from the floor here at Mass General where spinal cord injury patients used to be treated. But the title is really a jumping off point that is about some of the patients who met one another there and decided we don't want to live like this. Like we don't want to, sort of live on White Nine our whole lives, and really served as a springboard. Oh, are we still hearing me? Okay. For the independent living movement and for the Americans with Disabilities Act, which celebrated its 35th anniversary this year. So we'll have the filmmaker and some of the protagonists talking about the film afterwards. One note, since this film is going to be entered into festivals, is our event still will be hybrid like usual, but it will not be recorded. So if you're interested, please do tune in or come here like usual. And that's October 21st. And also tomorrow afternoon, registration for that and for our two other fall lectures will be live on our Eventbrite page. Now, to get to tonight's speaker, who we're so thrilled to have, Neal Baer, MD, MA, MED, is an award-winning showrunner, TV writer, producer, physician, and author, as well as senior lecturer on global health and social medicine at Harvard Medical School, and founding co-director of the master's degree program in media, medicine, and health at HMS. Here are just some of his TV credits. He was most recently executive producer and showrunner for the 3rd season of Designated Survivor on Netflix. Previously, he was executive producer and showrunner for the hit CBS series Under the Dome, the CBS medical drama A Gifted Man, as well as the executive producer of a show we've all heard of, the hit NBC series Law and Order Special Victims Unit from 2000 to 2011. And before his work on SVU, he was executive producer of the NBC series ER. A member of the show's original staff and a writer and producer on the series for 7 seasons, he was nominated for 5 Emmys as producer. Here's a notable film credit of his. In 2020, he was given the Special Jury Award at Sundance for his executive production of the film Welcome to Chechnya, and after it aired on HBO in 2020, it won the Peabody Award. I'm going to keep on going. He's also a writer. His first novel, Kill Switch, co-written with his SVU colleague Jonathan Green, was published in 2012, and he teamed up with Green again in 2015 for Kill Again. And the reason that he's here tonight is that he's also the editor of a 2024 book of essays about the gene editing technology CRISPR. And with that, we'll turn it over to Dr. Baer. Thank you so much.
Wow, that made me tired. Thanks so much for having me. It's a pleasure to be here. I haven't really been at MGH since I did my radiology rotation many years ago. And I was thinking about it because we were doing presentations and we were told to make it 3 minutes and we could only have one film. And so of course there were some students who did multimedia presentations and had many films. And so that became for Noah Wiley, who won the Emmy the other night for The Pit. That became Noah Wiley doing exactly what he was told, and Ming Na doing the multimedia project, and Noah's character, John Carter, really reflecting on how, when you follow the rules, you don't get anywhere in medical school. So it was, when I did ER, so much was inspired by my wonderful colleagues and the faculty. So I'm really excited to be back here tonight to talk about the promise and peril of CRISPR. So thank you for having me, Sarah. So what is CRISPR? So it stands for Clustered Regularly Interspaced Short Palindromic Repeats. And it's pretty simple in its concept, but of course it took years to do. It's a cut and paste system that homes in on a DNA gene sequence and snips it out using an enzyme called Cas9 enzymes. And they're developing newer and better enzymes as we speak, biotechnologists, replacing the original gene sequence, pasting it in with a new one that rewrites or repairs it. And by repair, there's a new system, which is base editing, where we can actually, not me, but these geneticists go in and change one letter in the DNA sequence, like from adenine to, or whatever, to guanine, whatever. So it's an incredible technique with obvious uses to treat genetic syndromes and some terrible genetic diseases. And how it started was some scientists who were working with yogurt at Danone noticed that some yogurt went bad because it was attacked by viruses, or other yogurt did not. And they noticed that the bacteria was incorporating pieces of the virus, and therefore the genome of the yogurt was able to recognize the virus in the future and mount a defense against it. So that was kind of the idea that, oh, you could cut up DNA and play with it in many different ways. So there are two kinds of cells that CRISPR can be used to edit. You can also think of it as like, sort of the old-fashioned word processing, definitely not texting on your phone, which autocorrects and makes mistakes. So that could be CRISPR too, in a way which I'll describe. But it's when you go in and you wanted to change a couple of letters where you made a misspelling error, then you could go back in and put the correct letters in. You had to find it. That's the messenger RNA. And then you had to cut it out with this, or delete it. And that would be the enzyme, the Cas9. And so, when you're texting on your phone and it auto-corrects, and it's not what you meant, well, there are oftentimes, in early phases of CRISPR, off-target effects, where even though the editing took place in one specific spot, there might have been off-target deletions or mutations as well. And there's been a lot of work to reduce that. So there are two different kinds of cells that we talk about: somatic, and that's from the body, and germline from gametes, the reproductive cells, and they can be manipulated using CRISPR. So we're going to get into the differences and why somatic versus germline is so important. So only germline genome cells can be inherited by future offspring. And you've certainly heard about Dr. He in China, who edited an embryo that then split into twins, and he manipulated, using CRISPR, the receptor for HIV. And so then he went to prison and paid a fine. And in our book, we talk a lot about Dr. He, and the last chapter of the book is really quite interesting in that Ben Hurlbut from Arizona State University, who's an ethicist, writes about how he attended some meetings where Dr. He was there, and he was encouraged by some scientists, he doesn't say who, to go forward and make China proud, and maybe he'll win the Nobel Prize, like the person who did in vitro fertilization, who didn't do it the traditional way and announced it through the news. So there was some interesting backstory to Dr. He in the United States, because he did his fellowship and postdoc in the US at Rice and at Stanford. And so Dr. Hurlbut argues and I think pretty persuasively, that oftentimes science leads the ethics. And it's, he makes the case that these scientists maybe had some ulterior motives to get Dr. Hood to do what he did so that it would be more accepted going forward. And I'll talk a little bit more about that in a bit. So if we can edit germline genome, that means that the future offspring can inherit whatever edit we made. If we do it somatically to just cells like, say, in the liver, which we'll talk about as well, they can't be inherited because we're only, say, editing liver cells or blood cells in the case of sickle cell disease. But if we edit the embryo, then that edit will be passed on in future generations, hence by using the word peril. So using CRISPR to treat human disease began in 2019 when scientists in the US used it to treat a patient with sickle cell disease. And that was successful. And it's a terrible disease. When I was doing my residency at Children's Hospital Los Angeles, I had a patient, a young teenage girl who had sickle cell disease, and she had terrible, terrible pain. Many of these kids have strokes. About 100,000 Black Americans have sickle cell disease in the US. It's estimated about 8 million around the world. So you can imagine if you can use CRISPR to treat sickle cell disease, you're really accomplishing a tremendous feat for humanity. It's expensive, and it takes a lot of time and risk for the patient in the hospital, but it has worked, and there are more and more people who are getting it. But it raises questions about who can get it, who's going to pay for it, and things like that. Though it's estimated that during a person's lifetime, which is much shorter with a person who has sickle cell disease, it costs about $1.6 million to treat them, with maybe $50,000 to $60,000 of out-of-pocket expenses. So it's a very expensive disease, but more importantly, it is a very, very painful, very harm-inducing, not, of course, for the child and the adult, but also for the family. So in Europe, scientists treated a patient with beta thalassemia, another what we call hemoglobinemia. And why is it that we can treat people with CRISPR who have blood diseases? Well, it's pretty simple. We can infuse them and we can hit their blood cells. It's much more difficult and challenging, and we haven't been able to do it yet, to reach, say, lung cells for cystic fibrosis or muscle cells for Duchenne's muscular dystrophy. But there's a reason why we can address genetic diseases in blood, because we can enter the blood. And why the liver? Well, blood goes to the liver. So we can deliver the CRISPR, the messenger RNA, and the scissors, the enzyme, more easily to those kinds of places in the body. So why should we worry about using CRISPR if it can be made safe in the clinical setting? So it appears that the research and all the work that's been done has shown that it is working for people, say, with sickle cell disease. And you may have read in the New York Times, maybe about 8 months or so ago, about a baby who was 9 months old who had a very rare metabolic disorder, 1 letter difference. And that child would suffer and likely die. And they used CRISPR to base editing to change that letter and hopefully cure that baby. And it looks like they succeeded. So, and that was the company, I think that was partly Vertex, who did sickle cell disease treatment, and others. So if we can make it safe, and there's still issues of off-target effects, but if we can make it safe, why should we worry about using it? Well, repairing the somatic mutations, the cells in the body, not the germ cells, means that scientists can do the same thing to the germline genome. So that's why we're worried about it, or I'm worried about it. This could change human evolution, if you think about it, by making an edit in an embryo that will be inherited by descendants. So evolution takes millions of years. You know, we think that sickle cell disease came about, and you probably know this, that if you're a carrier, and even carriers are not, there's this kind of myth, I think, that carriers have no effects from having one gene instead of both. But there seem to be some effects, but not like having sickle cell disease where you have both genes. So it's a 1 out of 4 chance because both parents have to be carriers. So there's a 1 in 4 chance that you will have it because you have to have both genes to have the disease, or you're a carrier. So it appears that through evolution, that carriers may have been more resistant to malaria. And consequently, that's why more Black people have sickle cell disease, because they came from that part of the world. And so it was protective, but evolution doesn't always protect everyone. And so even though you're protecting more people, because there are many more carriers than there are people who have the disease, evolution sacrifices those for the many. So that's the thinking, that's how that mutation came about, because it allowed more people to live and reproduce. So CRISPR raises the possibility more realistically than ever before that scientists will be able to rewrite the fundamental code of life. And that's Michael Spector from The New Yorker who said that. And I read this some years ago, and I got interested, both because I said I had treated this child with sickle cell disease and lived through many crises with her, but it, just so daunting, and many times where she almost died. I was really interested in what could work so that no child would have that. So let's start with the ways CRISPR is used, and we'll focus in ultimately on human beings in a moment. So the impact of CRISPR goes beyond the human species. Any gene can potentially be edited, whether in a person, animal, or plant, if it has a genome. So there have been lots of CRISPRing of animals, pigs, basset hounds. You can order, I'm not sure if you still can, but you could, the last time I looked, order online a kit to make giant tree frogs. And you could CRISPR the frogs, and they would grow because you could CRISPR the gene for their growth. And kids in biology in high school often CRISPR yeast and make them fluorescent. And that's really cool, and not terribly harmful, I don't think. But we have an article in our book about how far do we go with CRISPR? Should we just make chickens with even like very tiny heads that are just like meatballs? Should we make transhuman animals and keep going forward? Here at Mass General, they're transplanting, kidneys that come from pigs and that have been developed using various techniques. How far do we go? Do we make animals sentient at some point? What do we, what should we do? What shouldn't we do? I think are questions we need to ask. So researchers have successfully modified genes in chicken cells and made them resistant to avian flu. That's pretty cool. Could this kind of intervention inadvertently lead to new variants that target humans? I don't know. I worry about, thinking about past technological revolutionary innovations that changed our lives, like fossil fuels or plastics or nuclear energy. And yes, those things have made lives much better, but they have come with a cost that wasn't necessarily predictable when they were first coming out. We didn't know about microplastics then. We still don't know really about microplastics. We know a lot more about nuclear energy and nuclear weapons, and we know a lot more about fossil fuels and climate change than we did, of course, in the Industrial Revolution. So what I'm saying, which I think is pretty clear, is we don't know the future of CRISPR. We know how well it's doing now in certain cases like sickle cell disease or beta thalassemia, but we don't know how it can be used in the future, and that's not possible for us to know. But we do have to think about it so that we don't keep going down a path that's, regrettably not one that we can turn back from. So biohacking or hybridization has long been used in livestock, poultry, and agriculture industries to improve productivity and decrease the time it takes to raise animals, plants, and grains for food. So hybridizing is a way of selecting out genes. It's not CRISPRing them, not cutting out genes, but it's a way of selecting genes. With CRISPR comes new questions such as those raised, as I told you, by philosopher Marcus Schulz-Bergen. Will CRISPR improve animal welfare by decreasing suffering and improving health? That's an important question to ask because that would be a good reason to do it. Or will it lead to headless chickens or football birds that wholly lack consciousness, effectively becoming growing meat sacks? Now, it's interesting also, we're able to make cellular meat without killing chickens or cattle. We're even able to make milk, cow's milk. There's a company called Turtle Tree Labs that can make it, not to scale it yet fully, but can make it. So what is that going to do? Well, it sounds pretty good to me if you can make cellular meat that tastes exactly like beef, and you're not having cattle spewing methane, which is a huge component of climate change. So there, are many things that we can do that I think will help the planet. And then there are things that I worry about that we might do that will not. So when I did the show Designated Survivor, as Sarah said, I made the third season all about CRISPR. And so I am worried about the perils. We talked about germline editing, evolution. There've been discussions that are the Russians and Chinese thinking about or even doing CRISPR to make soldiers that need less sleep, because we do know the mutations that cause people to need less sleep. We know the mutation that confers no pain on children, which is a terrible thing, because they don't feel that they're getting burned or they don't feel that their bones are broken. Possibly some genes for fear are making us less fearful. So should we be making soldiers that can have these qualities? These are questions. And then, of course, with bioweapons, yes, we can make bioweapons without CRISPR, but CRISPR can maybe perhaps make it easier to introduce more transmissibility or new combinations of things. So I'm going to play a scene from my show that discussed, I think we're like one of the first shows that really discussed CRISPR. Here we go.
Dr. Mays?
Hold on. I'm trying to prevent the complete deforestation of Indonesia by increasing the genetic expression of algae to produce palm oil. Tribonema, Ulothrix, and Euglena have the best potential so far.
I'm Hannah Wells.
I'm also genetically experimenting with the cure for gray hair.
How would you like to come and help the CIA?
Hard pass.
Why?
I'm a biohacker. We're about the democratization of science. The CIA is not.
For a reason. Bioengineering in the right hands, great. In the wrong hands...
Exactly. Gene editing is listed as a weapon of mass destruction, right? You guys aren't focused nearly enough on bioterror.
Well, that's what I've been working on.
Mutually assured destruction is passé. Now, it's a bio-rogues world. The Biological Weapons Convention isn't enforceable or even verifiable. If you don't hold people to account, shit will happen. You need to be monitoring everyone, like I do.
How do you monitor everyone?
For one, I attend bioterror conferences all over the world. I give talks on things that keep me up at night.
Like what?
How easy it's about to become to make biological weapons on our phones. They'll soon have the capability of becoming nanotech assemblers, and when they do, there will be sequences you can print from your phone that can probably cause civilization to crash. And someone, either intentionally or accidentally, is going to do it. And without intervention, we're all ultimately screwed.
What worries you the most?
At this moment? This. Hundreds of them fell from the sky in Florida a few months back. They died from a virus I've never seen before. I believe it was genetically engineered.
For what purpose?
That's another thing that keeps me up at night.
Well, who should we be talking to?
I assume that was a royal we. Remember that hard pass?
You just said that the CIA isn't doing enough to monitor bioterror, so help us.
I know a biohacker with a lab near MIT who was experimenting with the avian viruses. Nolan Fine. He's not Fed friendly.
Then it's a good thing you're gonna be there to make the introduction.
Yep, he joins her. So where did this whole idea come from? It did come from someplace. So where it came from, and I was writing this in 2018 during the first Trump administration. So has anyone heard of Project Coast? So I'm going to talk about that, I think, in a minute. So Project Coast was a biological warfare experiment done during apartheid in South Africa. And the idea was to design some kind of biological weapon that would target black-skinned individuals. Fortunately, it never happened, but that got me thinking, what if CRISPR could be used in the same way? And I was thinking about the great replacement theory and things like that back in 2018, and we kind of are stunned that we were somewhat prescient in this show. So we were thinking, and this is a spoiler, but the whole, why is there a cardinal? Well, I love cardinals, but it was a story about trying to figure out how you could design CRISPR to attack melanocytes, or the cells that cause skin color. And so they started it, the people who were doing this started it with, the color in cardinals, and then we're planning to move it on towards people of color. Because there was a concern that there were too many people in the United States of color that would outnumber people who were white. And it's lo and behold, we hear about this a lot now. And so, that was the thinking behind doing the CRISPR story. And this is the very beginning of that storyline. Can it happen? We talked to experts. And we talked to experts from Johns Hopkins, and they're like, why are you asking us these questions? And it's like, no, this is for TV, because I'm interested in Project Coast, because, there are people in the administration who actually came from South Africa and have some history with apartheid. And I'm interested in these things. And so, or now they're in the administration. They weren't back in 2018, but that's the prescience of it. So, whether or not it can be done, I don't know. But certainly CRISPR can be used to target certain kinds of genes, and, skin color is made with various genes. So I wanted to, and ultimately what happens is what they were doing was looking for the skin color genes and then making these people infertile so that the numbers of those people would be much lower. So that, vis-a-vis the great replacement theory. So, so that's how this TV story came about using CRISPR. I also talked to someone from MIT about gene drives. So conceivably, a gene drive can be made to affect, say, mosquitoes. They work well in animals and insects particularly that reproduce very quickly. And so you could conceivably get rid of Anopheles mosquitoes that cause malaria. The problem is that we don't really know what the impact, do we truly know the environmental impact that mosquitoes have, those bad mosquitoes, on the world? I mean, they are here for a reason, and they do things to us, but we're all animals. And so is it prudent for us to wipe out Anopheles mosquitoes. There's a really good piece in the New Yorker some years back where they talked about doing this on Martha's Vineyard to eliminate Lyme disease and getting rid of the mice, the deer mice that carry, the ticks. And so the community voted not to do it. But there was the real strong possibility that using this gene drive that causes them to be infertile, that they could really get rid of Lyme disease. So these are things that are, really with us and possible that we need to, as citizens, talk about. So the babies that I talked about before. So Dr. He, a Chinese geneticist, used CRISPR to manipulate the germline of embryos that became twins, claiming to have made them resistant to HIV, and an uproar ensued. And we're really not, he said he did this because the father of these twins was HIV positive, and there was, rampant discrimination still going on in China towards people with HIV. The problem is that for many, many years, we've been able to help people with HIV, both the sperm donor or the egg donor, not transmit the virus to their offspring. So you don't really need CRISPR to do that. You don't need CRISPR to do, say, for instance, you want to get rid of sickle cell disease. You could test people and do in vitro fertilization, and we call it pre-implantation diagnosis, where PGD, where we look at the fertilized egg and we only implant the egg that doesn't have the fertilized egg that doesn't carry cystic fibrosis or sickle cell disease or Tay-Sachs disease or Marfan's, et cetera. There's a long list. So you don't really need CRISPR to necessarily prevent genetic diseases, though there are geneticists who say it's better just to get rid of these diseases by doing CRISPR on these people who have it, who are carriers, so that they don't have it anymore. So at the end of 2019, Dr. He and his two colleagues were sentenced, as I told you, to prison and paid a fine for knowingly violating China's regulations and ethical principles with their experiments. The court indicated that 3 genetically edited babies have been born. And we don't know really much of anything, I don't think, about the 3rd baby. And these babies, apparently, we don't know that much about them, were mosaics. So they don't have, every single cell of theirs does not have the resistance, doesn't have the mutation. So let's talk about CRISPR and diversity. So how one defines a disease or a medically unmet need is influenced by the culture and those in power, including the medical industrial complex that is driven to find cures, because they do make money. It is expensive to do it, to do the research, and it then costs a lot to get the treatment. So Dr. Carol Padden, who's deaf, and she's at the University of California, San Diego, and is the Dean of Humanities there, and her daughter, Jackie Humphreys, who is not deaf, point out that many in the deaf community do not see themselves as disabled, but as valued contributors to human diversity, with a highly developed language and style of communication. So she sees herself as part of human variation, and therefore does not need to be fixed or cured. Now, there are people in the deaf community who take a different approach. This is epitomized by the debate on cochlear implants. Some people who are deaf have them, like Marlee Matlin, who's a well-known Oscar-winning actor, and there are others who don't. And we did a show on ER, actually, where I interviewed doctors who were deaf, some who had cochlear implants and some who didn't. So we had Marlee on the show, and we had Phyllis Freelick, who played that same character from Children of a Lesser God on Broadway, and she did not have a cochlear implant. So we were able to get at all of the issues, which I love to do on my shows, of all the points of view about why some people do it and why some people do not. And why am I bringing up deafness? Well, deafness is really interesting in that I just was telling you about PGD, pre-implantation genetic diagnosis. Well, deafness is an unusual case, or very, very unusual case in that pre-implantation genetic diagnosis is not going to work if both parents have a genetic deafness and they have a child and their child will be deaf. And there's no way that child will not be deaf. For instance, with sickle cell disease, it's, you need both parents to be carriers, or one parent can be have the full disease and have both genes. But with some genetic diseases, the parents, will both have the genes, and it's not uncommon for deaf people to be in deaf communities and have children. So some scientists are now saying, and some physicians, that this is the unusual case, but there may be a time to do germline editing, because that's the only way that this couple could have a child who is not deaf, if they chose to have a child who is not deaf. So this is like the open door. Like in 2015, the academies all were saying, no germline editing under any circumstances. And then after Dr. He, we started to hear about these exceptions, these cases. So I worry, like when we go with the exceptions, we're starting to go down that slope. Like what's the next exception? And, and once you do something, it makes it easier to do it again. And that's what Van Hurlbut argues in his piece that I spoke about, is that science is leading the ethics. Because at first it was under no condition, and still it's not legal to do germline editing. But how do I know that it's going to not be done by some rogue scientist like Dr. He? So I'm going to show you the post-apartheid piece that I was telling you about, and what went on in our show in discussing what was behind the case of that cardinal.
You're certain of this?
On a hunch, I rechecked the samples from my bird friends and discovered a similar pigment targeting mechanism.
Wyatt was only targeting redbirds.
Are you familiar with eugenics?
Yeah, Hitler, Nazi Germany.
And Long Island, and an American doctor, Charles Davenport.
He studied heredity badly.
He's the father of a scientific movement. Promoted the idea of racial genetic superiority, very popular at the time. Fans included Madison Grant and Dr. John Kellogg.
The cornflakes guy?
Yes, and Hitler, who used Davenport's work as a blueprint.
Not our proudest export.
But one of our more enduring. Take apartheid South Africa.
Project Coast, the bioweapons program.
Yes, I was assigned to the team to monitor Project Coast. They were researching ways to genetically target black South Africans with lethal diseases.
Did they succeed?
No, thank God. But their work, like Davenport, continues to inspire.
Yes. So, things to think about. I think the point here about eugenics is important because, there are a lot of diseases around the world, and USAID really no longer exists. So what is that about? Why is that? And I think that this idea that survival of the fittest is how we should go about the world in dealing with people around the world has really taken hold. And, you can think about that, whether you agree or not, but it seems to be more sort of in the air now. And for me, quite frightening, because people who are exposed to disease, it's not really about their fitness per se. It's about whether or not they can get vaccinations or live in places that are protected from, mosquitoes or bacteria or viruses. So for CRISPR, the pros versus the cons. So let's go through. So it's amazing, can cure genetic diseases. We know that. And I think that's fantastic because, as a physician, pediatrician, I don't want children or adults to suffer. But the con is potential for biological weapons. And, these DNA sequences that labs and scientists can buy are not really monitored. There's no real law that's looking at who buys what and who's getting access to them. So I think, scientists may argue that they can oversee their own. But then I give this example when I talk to scientists who say that, and I say, have you ever jaywalked? And they'll say, well, yeah. Have you ever known anyone who's driven even slightly inebriated? Well, unfortunately, yeah. And I said, well, there's laws against that, but people still do it. They break the law all the time. So you have laws, I think they're important, but how do we enforce them? So how do we enforce a moratorium on germline editing? So another pro is advances in gene editing. It's getting much better with fewer off-target problems, but there are ethical concerns, as I said, about maybe who would get these. What if we do enhancement? Who's going to be able to pay for it? Michael Sandel, who's a professor in government at Harvard, wrote a piece, very, very prescient, I think around 2004, for the Atlantic Monthly, and talked about, what does it mean to be human? And what does it mean to control your, evolutionary destiny? Now, some people will say, oh, you're overblowing this. It's like, but am I really when, you can target any gene and we can't change eye color with one gene. Eye color and whatever intelligence is or musculature is a combination of genes. And fortunately, fortunately, we can target many dire genetic diseases, and that's where CRISPR comes in. But there are ethical concerns about, as I said, bioweapons and who has access to the material to make things. There are a lot of DIY labs around the United States. We have a really interesting piece by Ellen Jurgensen, who's one of the first people, I think is the first person who started DIY labs to teach people about CRISPR. And I'm not saying that it's so easy, it's not, to go into a lab and do it. But, do I trust all scientists to do the right thing? No, they're people, they're humans. And so they jaywalk. I think we have to really talk about these issues and think about ways to have oversight over access to these materials. So the potential to eradicate malaria is incredible, but it's unpredictable long-term effects. Like, yeah, that'd be cool. That'd be great. There's so much suffering. There's so many children's deaths. But what does it mean to do something like that? How can we be sure that we're not doing something that opens Pandora's box that's not, fixable? Can treat RNA viral infections, regulatory challenges. Well, that's happening these days, and modulates gene expression. And we can do that, and germline editing risks. So there's pros, there's cons, but I'm not against CRISPR editing. I'm not against somatic editing. I do want to make it available to people. It is very expensive. So will Medicare, Medicaid pay for it? That's a big question going on for sickle cell disease, because there's, as I said, 100,000 people who have it with a often 20-year lower life expectancy. So these are, the regulation of CRISPR. So safety training and licensure is important. The ethical considerations of doing these things, germline editing, should we start doing this with, families who are deaf, who don't want a deaf child, and then what's next? Research and innovation, how do we oversee that? And justice and equity, who gets it, who doesn't, who pays for it? So some questions to consider while I kind of finish up here. Where will the CRISPR path lead us? Is it possible to reconcile the dual use of CRISPR technology? So I like this term, the conundrum of dual use technology, dual use. So that means there's the positive, curing horrible diseases, and the potential for, disaster with unforeseen results, bioweapons, enhancement, things like that. So do CRISPR's benefits in medicine and agriculture outweigh its risks of potential weaponization? And Have any of you heard this discussion going on? I doubt it. You don't hear politicians talking about it. So reproductive and gender issues. So where will the CRISPR path lead us? So how might we understand a mother's reproductive freedom versus a fetus's future health? Should CRISPR technology be used to enhance one's physical desires, say, in the case of trans individuals? So we have a paper on this where, might we be able to give people CRISPR genes so that they don't need to have exogenous hormones? So that's a good question. Should we do that? Should CRISPR be used to make a biological child related to both gay or queer parents? All right, so this is possibility. There's also down the road another technology called IVG, in vitro gametogenesis. Has anyone heard of that? So IVG was developed in Japan. There's a great piece in the New Yorker about this, where scientists took a mouse tail and they coaxed the pluripotent cell to become a sperm or an egg. So what does that mean? This is the world of Lysistrata, without any men. So it's conceivable that you could do that with, say, a woman who didn't really want to be involved with a man, or had, let's say, a lesbian partner, or on her own. So she could get a sperm cell from her partner, her female partner, let's say, if it was taken from a pluripotent cell. Now, it's not legal to do that, but it's down the road. It's being researched. So how do we recognize humanity in the, how can we even conceive of it in the future? Is really hard. I can't imagine these things, but it is possible. I mean, I can kind of think about it. I guess I can't imagine it because the basic technology is kind of there to start making it happen. And I'm not saying that we're doing it yet with people, but we are doing it with animals and looking at how to do it. So IVG, if you're interested, you can look it up and read more about it. So where will the CRISPR path lead us? Who should have authority over decisions about whether, when, and how CRISPR should be used? Scientists? Politicians? Academies? Hospital? How might we oversee who has access to CRISPR to prevent its misuse? So what could possibly go wrong? All right, so this is really an interesting story. This doctor, Catharizin, has edited the PCSK9 gene to permanently inhibit that gene, reducing LDL cholesterol by an average of about 60%. And this is known as one and done gene editing for a one-dose future. So let me tell you about this. So PCSK9 is a gene that controls your LDL. And the way it works is that PCSK9 degrades the receptor for the LDL, the bad cholesterol, so that it goes into your blood. You need some cholesterol, but not too much. And some people have mutations where they have they have too many degrading receptors. So that's bad because it puts more and more LDL in their blood. And so they wanted to, this doctor at Vertex wanted to make a way to treat people who have hypercholesterolemia, which is not terribly uncommon in the United States. And these people have very high cholesterol, and they don't respond very well to statins, like to Lipitor, and they don't necessarily respond well to the injections or to the antibodies, the monoclonal antibodies. And so it was thought, what if you looked at this gene and you turned it off? What would happen? So with CRISPR, because it's in the liver, it's easier to reach than like a muscle gene, a bone gene, a lung gene. You could go and deliver to the liver cells using the messenger RNA and then the enzymes, and you could take out using base editing, just like take out a letter and do the editing so that the person would not have their receptors degraded. And that's how they had the 60% decrease. And it's so important because these people are at such high risk for heart attacks, having heart attacks in their 30s. And so this was amazing. And so there was a lot of discussion. They've been online with people who've had it. They infuse it one time, 4 hours, and that's why it's called one and done medicine. Like, you know, you got to take Lipitor every day or get the Repathin shot once a week or every 2 weeks. This you get once and you're done. That sounds pretty cool. All right, so let's see, how do I want to go to, I don't have the mouse here, but let's see if I can, whoops, do this. Is there a way for me to go to enrich KRG? I forgot that I needed to show you. Thanks.
You can just use the mouse.
Oh, it's right here.
Yep.
Perfect. Thanks. All right, so I'm going to put in here, this is an AI platform called Enricher KG. And thank you to Barbara Natterson Horowitz for telling me about this, who's a professor at Harvard. And so I'm going to put in PCSK9 right here. And I can select from libraries. So I have a biological processes, mammalian phenotypes, and I'm just going to look and see. And here we are. So PCSK9 doesn't just do LDL. It does, and it's hard to kind of to see this, but it does decrease circulating LDL, increase bile salt levels, catabolic processes, negative regulation of lipoprotein particle clearance, negative regulation of low density lipoprotein, does some things with sodium ion transmembrane transport. So my point is that, yeah, it's great to do this one and done, but these genes have been around for millions of years. In fact, this gene was found in acorn worms. So it's like early life. And it's evolved over, not the gene, but the gene is found in primates, human beings, of course, rabbits, but not in bats or cats, and reasons for which we don't really know. But we do, there's been some speculation that possibly you need this gene. If you knock this gene out, it could lead to sepsis. This is, I'm quoting from an article in Circulation from 2024, possibly metastases. It may control these things, but they kind of say at the end, but we won't really know until it's done. So, okay. So my point is that, you know, in the discussions I've seen online about PCSK9 and the one and done, I just want to be sure that we're also clear that this gene doesn't just do one thing. And by knocking it out, yes, we can help people who have really high risk for heart disease and heart attacks. But we just have to be really clear that we're not doing something that may have dire consequences down the line. So I find this really helpful to look at genes that are being edited to understand where these genes came from and what they control. So I'm going to stop there and take questions.
All right, thank you so much. Let's do a round of applause first. No some questions yet. All right, I'm coming your way.
Thank you. Very interesting. My question is, I understand, well, I don't know the answer, but I understand the issues about the ethics and, you know, unforeseen implications, etc. But assuming that it's decided, you know, that something is not good, what's the practical control? In other words, you said they were doing yourself labs, so I'm assuming it's not, you know, For example, we look at things happening in the world today, people say, well, do or don't send arms to these people. So that's obviously something that has a clear immediate effect. What are the practical aspects of CRISPR?
In terms of control,
Well, assuming people decided that they wanted to control, how could
they do it?
What's the feasibility?
Well, it's tough because right now they're really, there are laws against doing things, but people do things against the law all the time. So, I write about this in the book and I say a lot more conversations. People just don't know about CRISPR unless they have sickle cell disease or something that's very specific where they can be helped. But these are conversations that need to be had in schools, in colleges, in communities, in churches, but we just don't have these conversations. And I think I've talked to the American Association for the Advancement of Science about this, is that we really need to help scientists tell their stories better so that citizens and communities can understand what's going on and ask questions like you are, because I don't have answers. I mean, there are things that are being put in place, like supposedly companies that make DNA sequences are supposed to report if somebody's ordering some pieces that might, we know the DNA sequencing of smallpox and Ebola and things like that. So if people are ordering some things that look like they could be, used for something, nefarious, it's incumbent on these companies to, report this to the FBI, which investigates these things. And so we need to have laws that are very clear that you can't order, that we have to, you have to have gone through training. I mean, we have chapters about this, but it's very complex. But we do have to inform the public, because this is where we're in a real chasm. Like, the public doesn't really understand science, and they're not liking scientists, and they're afraid of science, and looking for home remedies like cod liver oil to treat measles. I mean, so it's like, we look at here at MGH, at HMS, we look at the data, and the data inform us. What's tricky is that, for instance, COVID is not, what we know about COVID is not definitive. What we know today is very different from what we knew 4 years ago. We didn't really know much about long COVID. We're learning about who's susceptible, why they're susceptible, what genetic profile they have that makes them more susceptible. And so people want science to be definitive, and it's not. And so we have to help people understand that science is an accrual and a process of accrual. We're learning, we have a theory, we test it, and we move forward with the truth from what we have found in the experiments. But if we don't understand that as a community of citizens, and we're just suspicious, then we have a long way to go. So we have to work with schools, but it's a very challenging time, of course, with RFK and anti-vaccination and anti-science. It's anti-data, anti-empirical research. It's a very troubling and challenging time, but we have to speak up, and we have to say that we need to teach young people about scientific method. You're asking one of the hardest questions, like, how do we prevent disaster from happening while we still have the opportunity to do that? So we need to talk, and you need to, talk to people and have conversations with scientists who do this and hear what they have to say. I've talked to scientists who say, "Oh, we can oversee our own." And I was like, okay, but that doesn't make me that comfortable. You're going to oversee your own, and we'll make them pariahs. It's like, come on. I mean, so we do need some conversations that are, not about trans people harming us, but about CRISPR and what the consequences are that are based on the data.
And you don't have much data.
On what? On CRISPR?
You haven't had so many
Well,
experiments.
Exactly. We don't have, people, some people say that we don't really even know the effect of IVF because people haven't lived long enough. But it seems that the longer they live since the first baby, we have pretty good information. But we don't know, until we don't know about long COVID until people have had it and we see it and we can identify it and we can analyze it and experiment. I used to say, well, we were pretty definitive about measles. And then I read that some Harvard researchers came up with, like, why do children die from the measles? And the answer seems to be that measles can affect the antibodies of children. And if your antibodies are affected, then you might not be able to fend off pneumonia or other things. And so we didn't really have a clear idea about this until this year. We just knew that we knew like epidemiologically the number of kids who have measles who would likely be hospitalized and who would likely die because we have big data on that, which we don't necessarily yet for COVID. So we had that information, but now we learn more things. So science is not definitive. It is ongoing search for the truth. And that's very hard for people because they want to know what caused autism in my child. Well, autism isn't one thing. Autism is now a spectrum, and there's pretty good arguments that we have higher rates of autism because we define it differently. But also, one child who has autism is very different from another child who has autism, and they may have very different genetic profiles. We have some ideas. It could be some environmental, could be parents' age, but we do know from the best studies that have been done that it's not from vaccinations. So, how do we help people understand, science? And I think one of the ways is having conversations and getting away from, back to the first question, getting people together to have conversations like, well, we're having kind of a conversation, but we're talking about it and you can ask questions. That's why I said that I said to AAAS, climatologists should go into Starbucks and they should sit there and talk to people about, who want to talk. Because we do know from the research, that's the best way we have, that when we get into a situation where people can converse and have a conversation, that there's a much better likelihood that they will have a better understanding. And we're not there to shame them and to make fun of them or to say that they're stupid, but to listen, understand where they're coming from, and then present the data and the stories that go with the data.
Right. So, question,
She's going to bring the mic to you.
so short question, COVID-19 certainly changed for the worse by leading to death of tens of millions of people. A lot of people are thinking about settling the original COVID-19, which came out with Wuhan, China, actually government lab. Have you heard of any evidence for anything or any suggestion that the original version of COVID-19 get killed?
Well, I've certainly heard lots of conversation about it, and there's lots of debate about it. It's a tough question in the sense that, do we have all the evidence? Like, do we have everything from China so that we can look and see what happened? So I think we need to have the evidence. So I can't comment on whether or not it was made with CRISPR in a laboratory, or if an animal, if a bat or another vector animal passed it on through the marketplace. I do know from what I've read that there were bats that had a very, very similar virus to COVID that they knew about some years ago, and they were looking at the DNA of these bats. But importantly, I think we have to be really open about the data, Because if we don't have access to the data, then we have conspiracy theories and no, I can't prove or say that it didn't happen or it did happen. But I need to know what happened in that lab. And if I can't have access to that lab, then I'm not, so it's a good question in the sense of transparency and being transparent about what's your intention. Like, is your intention to cure disease, like sickle cell disease? Is your intention to enhance people? What's your intention? Do germline editing, and because you can charge a lot of money because you're claiming that you can confer genetic superiority over with this, CRISPR technique, there you can go to some physicians who give screenings for certain attributes, genetic attributes that, your child could have, and people go to sperm donors, let's say, or egg donors for certain attributes, but it's not so specific as CRISPR. So I think transparency is key, but first and fundamentally discussing these issues.
Okay, ask a question now. Guess you, Curtis. How did this book project come about? Were you approached, or were you interested in the topic and you wanted to sort of pursue it and gather all of these perspectives?
So it was from me. So because of the patient I had at Children's Hospital, Los Angeles, and she almost died one night because she had gotten, this was in the days still where we had like files, and buried in an old file was don't transfuse her. And she had a transfusion reaction because the team didn't know, because we didn't have everything on computers. And I was, the intern, so I was like reading like all night, like every file. And it's like, oh my God, she was transfused and she almost died, but she didn't. But that's where I was like, wow, this child has suffered so much. And I talk a little bit about the cultural manifestations. Like a lot of these children and young adults and adults are in such pain and then are seen as drug seeking. Well, I think they are drug seeking because they're in so much pain that even our best, drugs don't often relieve that bone pain that they have. But they're minimized, and it's really been terrible for kids and people with sickle cell disease. So that inspired me, and I read about it and learned about it because I was interested in it. And then I'm on the board of, I'm on the editorial board of a journal called Perspectives in Biology and Medicine that's published by Johns Hopkins University. So we did a special issue where I gathered some papers and wrote the introduction, and our dean from Harvard wrote the introduction, I wrote the commentary for it. And so I thought, wow, this was in like 2019 or 2020. And I thought, this world of CRISPR is growing and evolving quite rapidly. It would be interesting to expand it and make it a book. So I went back to Hopkins because they published the journal. And I said, what if I add like eight or so more and take out some and add some new things and talk about COVID and update it? And they said, yeah. So that's how it got made. And then, of course, I did it on Designated Survivor because I'm always looking for ways to interject the science into the work I do, the science and the medicine.
I have like two questions. You talked about the somatic editing being one. Is that passed on when they have a child in somatics, just with them? No.
Just with them, because it's, for sickle cell, actually the way it works is they turn on fetal hemoglobin. So when a fetus is in utero, it has different hemoglobin than it does when it's born. So when it's in utero, it needs a hemoglobin that can really hold on to oxygen because it's not breathing, it's getting oxygen from its mother's blood. And when the child is born, then that turns off and the other hemoglobin comes, your hemoglobin comes on. And so these children are okay when they're fetuses, and then they're born and then trouble starts. So when we somatically use CRISPR, we're addressing cells, but not the germ cells, the reproductive cells. So therefore, just the cells that, you know, are involved with sickle cell disease, or like with PCSK9 and LDL. We're not doing things to other, hopefully, to other genes. We're not doing off-target things. So somatic changes can't be reproduced, but germline can. So that's why if we went into an embryo and it had sickle cell disease, because it had both genes, we could go into that embryo conceivably and change it. But there's no reason to do that, as I said, because we have pre-implantation genetic diagnosis, and we can look at the eggs and we can implant the ones that do not have sickle cell disease. Where things got complicated, or, you know, more complex, is that researchers started to think of the unusual cases, like the deafness case, where, okay, I have this egg, and every egg that's fertilized is going to have produce a deaf child. There's nothing we can do except use CRISPR to change that. And that's the beginning of, well, it is okay. Because in 2015, it was not okay under any circumstances. Now it's like, maybe there's a case or some different cases that are so unusual, it's okay. And that worries me, because then that makes it's always easier once you do it to then go forward and to go forward. So somatic is not, you don't pass it on. Germline you do. Like those twins, if they have babies, they'll have babies that their descendants will have, mutated receptors for HIV. Now, another thing that's interesting about that is there's some evidence, possibly, I've read, that while people who were exposed to HIV during, now or whenever, when they're exposed to HIV, they, didn't get it because they had the mutated receptor. Well, it may be that you're more susceptible to some other viruses if you have the mutated receptor. So evolution works to conserve life and make life continue. And so when we start playing with things and doing that, like with the PCSK9, I worry about that, because evolution has been working on it for millions and millions of years. Should we play with it?
Well, how can it then say that it's somatic, that we just say, oh, we don't see anything now, so it must have, what if it's kind of delayed?
Well, that's another question too. It's like, that worries me as well. Like, what happens with sickle cell disease? Or I was just saying that with PCSK9. It's like, okay, one and done, we knock out that gene and your cholesterol goes down 60%. That's really good. But that gene does do other things. And are we sure? I mean, I don't know. I haven't seen anybody talk about that when I've watched online conversations about it.
I don't know how it's much for a quick last question. Are they doing studies on, like, can you say turn off the gene, but then say environmental conditions change for humanity and that gene be turned on again? Have they turned it on or turned it off and on?
Well, I don't think there've been a lot of cases of CRISPR turning on and off genes, because it's not so easy to, it has to be a gene that you can reach. That's why blood or liver. And I don't know in animals how changing genes to make pigs meatier or whatever, or chickens meatier, what that impact is with the environment. And we haven't even talked about epigenetics, which is, another whole area. And so, so I don't know, certainly I don't think in people, we've done enough with CRISPR yet to even, we haven't turned things on and then off. I mean,
No one's ever really done that. So
not that I know of, but I do wonder being a writer, a TV writer, I do wonder if we can like use CRISPR to introduce like in some digestive way, the ability to make people like cows so that they could eat grass. Because, if we run out of wheat and things, like what if we could eat grass or bark? So I do wonder about, and then we could do that on germline cells, and we could make human beings able to maybe need less water. Like we're doing all kinds of research on making plants need less water for obvious reasons. Well, what if we knew what genes cows have? I mean, I'm sure we do know what genes cows have that allow them to digest grass. Is there a way to put that into a human being so that we could start eating grass and maybe start eating tree bark and leaves? Then we would maybe not be in such danger. Can we modify people? This is one of my shows actually. Can we modify people to, instead of changing the climate, because we seem not to be able to do that so well, can we change people? So I am interested in that. Can we modify people to meet the climate challenges?
Well, on that note, let's thank Dr. Baer again.
Thanks. Thank you for having me.